Hello, I am Kristin Landis-Piwowar. I am a medical laboratory scientist trained as a cancer biologist, and my PhD is in cancer biology. So I have some background in both the ability to perform laboratory testing and understand some of that, the necessity to have laboratory testing, and also to interpret it very much from the cancer biology world. I'm an assistant professor at Oakland University in Rochester, Michigan, and I'm here today to talk about, very specifically, molecular diagnostics in cancer and how we can use those molecular diagnostic tools to not only just detect, but also to help diagnose and use prognostic indicators as well. First off, I'd like to start this discussion with talking about carcinogenesis. I know that this is a varied population of people taking part in this bioconference, and I know that I've got people on all ends of the spectrum. So some of the first few slides may be remedial, or they may be on the advanced side, depending on where you're coming from. A side note, I am getting over a little bit of a head cold, so if I start coughing a bit, I do have my cough drops handy. But that's the beauty of doing these online talks, right? We can keep our germs all to ourselves, so my virus stays with me and all of you get to stay healthy. Regardless, when we're talking about carcinogenesis and discussing how a cell actually becomes a cancer cell, this is an important point when we start talking about the diagnostic issues and the laboratory testing that we can do, because we need to be able to identify how these cells are actually having been transformed into a cancer cell. So carcinogenesis, also known as creation of cancer, is a process. And it's a process of normal cells being transformed into cancer cells. There are three phases to this process of carcinogenesis, starting with initiation. A cancer gene is going to somehow be altered in the process of initiation, and this alteration is going to either be a large chromosomal change or an individual nucleotide or a small region of nucleotides that is altered. When this event occurs, if we don't have the appropriate DNA repair mechanisms in place to fix this particular alteration, then it stays within the genome of that cell, and then any subsequent progeny of that cell. And so we say that this is an irreversible event when we have initiated that cancer cell by causing some kind of alteration. The next phase of carcinogenesis is promotion, and promotion essentially leads to the clonal expansion of that cancer cell, meaning that that cell is able to multiply, it has mitotic capability, and can go through multiple divisions to show that we end up seeing that same mutational change that occurred during the process of initiation in subsequent cells. The environment of the cell has to be perfect for promotion to move forward. In that, if you change the environment of the cell, take away the factor that has allowed for that cell to be able to multiply, then you have stopped the ability of promotion. So this phase of carcinogenesis is reversible. The last phase of carcinogenesis is progression, in which we can see accumulated changes in cancer cells, multiple mutations, chromosomal instability. And once a cell has achieved this last phase of progression, it has basically fallen into an irreversible mutational event, and it cannot go back and revert and have any DNA repair mechanisms to fix what has happened. So at the bottom, in this lovely image, you can see that we start with an initiated cell here in orange, and that initiated cell has acquired some kind of a chromosomal change or a small mutation. If that cell is allowed to multiply, then we can see many copies of that in the process of promotion. And then we have a cell that has acquired additional changes which allow for that cell to then go through the processes of progression and make many, many, many more copies of itself. Ultimately, that can lead to a metastasis. This is pertinent to both hematologic malignancies and solid tumors. And so this talk is going to follow both hematologic malignancies and some of those tumors that are associated with organs as well. When we're looking for these changes that are occurring during this process of initiation and ultimately what leads to progression, what is being assayed for are chromosomal alterations and DNA alterations. Those are our two flavors for the genetic changes that are going to be seen. The chromosomal alterations are either going to be unbalanced or balanced, and I'll describe what that means in just a moment. And the DNA alterations are going to be reflected in many different types, but single nucleotide changes, small insertions, or small deletions, generally speaking, are what is seen when we have these changes leading to carcinogenesis. First, I will describe some of the chromosomal alterations, many of which you're probably familiar with. But a chromosomal alteration can be categorized as either an unbalanced or a balanced change to the overall genome in that particular cell. We can see large regions that are either deleted or duplicated. In either of those instances, those would be considered unbalanced chromosomal changes because you had either gained or lost a large portion of the chromosome, of the total genome. And so the total genome is now unbalanced compared to how it was previously. Alternatively, we have balanced chromosomal changes in the categories of, generally speaking, inversions or translocations. The inversions and translocations, generally speaking, are allowing for the same amount of total genetic material to remain present in the cell, but its location within the chromosome has changed. And because of that chromosomal location, what is created are new combinations of DNA sequences leading to new combinations, potentially, of genes and how those genes are going to be encoded for. So some examples of this are, as an example on the top here, you can see that there is a deletion that has occurred. If we have this total chromosome here that's passed from showing all of these different regions on the long arm and on the short arm of this chromosome, then you can see that over on the other side, we've lost some region. We've lost, my arrow's a little bit off here, but we've lost this region between genes 3 and 2, or these regions of genes 3 and 2. We've lost that region. It's a deletion. It is an unbalanced chromosomal change. A simple translocation is where there is just going to be one region of one chromosome here on this end that is going to, and on this end as well. There's going to be these two regions that are going to flip-flop. Or rather, I'm misspeaking myself. With a simple translocation, we just have this one region on the end moving on to the other end of one other chromosome. A reciprocal translocation is when the two regions flip-flop. I apologize for my misspeaking there. So here you can see that this region, this short region on the end of this chromosome, and this somewhat larger region on this chromosome have changed locations, as opposed to one large region moving to the end of one chromosome. In either case with translocations, also with inversions, there is just a change in the location of information, but all of the information for that cell is still present, whereas on the top with our deletions, there's actually a loss of genetic information. So deletions are only problematic when we're considering tumor suppressor genes. What are very common in solid tumors is that we see duplications. And so we can end up seeing large regions of one chromosome being accidentally copied, if you will, and having multiple alleles show up of one particular gene, where we were only supposed to have expression at one level of that gene. By having multiple duplications or multiple copies of that gene, we can end up seeing much higher gene expression, and that can promote a cell towards the progressive phenotype of what we see in the last phase of carcinogenesis. So with those translocations, if it's balanced, then we have this, what we're seeing is one segment of our chromosome is being attached to another. If it's a reciprocal, then we have an exchange between these two regions. How this occurs with these translocations is that there has to be some kind of a chromosomal break, and there has to be some kind of mechanism of DNA repair to have happened to actually fuse those ends of chromosomes in those regions. So again, this is not a change in the total amount. This is just a change in the position within the genome. So here you can see with this example, chromosome 22 and 2. If you're thinking along the lines of a hematologic malignancy, you could substitute maybe chromosome number 9 here, if you've ever heard of that translocation. And we might have some sort of an environmental agent that is going to, something exogenous to the cell, is going to be causing these DNA double-strand breaks. When these DNA double-strand breaks occur, we end up having these reactive ends to the chromosomes. And those reactive ends are problematic because if they're not put back on in the exact same position, they can, DNA repair mechanisms can recognize these as just being purely reactive and, oh, we need to fix this. And in doing so, what may happen is we have non-homologous chromosomes with different ends that were originally supposed to be there. The total amount of the genome stays the same, but what has the gene sequence, the DNA sequence, has now been altered and moved from one chromosome to the next. On these chromosomal alterations, the ultimate effect of having inversions take place, having reciprocal translocations or simple translocations, what happens is we can have one of three different things as the outcome, essentially. Fusion genes, a position effect, or a breakpoint effect. A fusion gene is where there is going to be some coding region of one gene connected to a coding region of another gene. And when those two coding regions come together, that creates a new function for that gene, that DNA sequence. And so this is considered to be a gain of function mutation by having these translocations or inversions take place. When we have a position effect, there is just a rearrangement of some of the regulatory elements that are allowing for gene expression to take place. And a breakpoint effect is where there is a loss of one particular region or another. This will come into play, and you can see how all three of these effects at the chromosomal level will play out when we talk about oncogenes and tumor suppressor genes, and how certain genes are going to be increased in their expression while other genes are going to be decreased in their expression. On the top, you see that we have a position effect in that there is the coding region for a gene, and having followed a simple translocation, a reciprocal translocation, or an inversion, what has happened is by flipping some of those DNA segments around or moving them to other places in the genome, one gene, in this case gene B, the coding region, as indicated by these sort of larger, thicker rectangles, that's the coding region for the gene, is now going to be in proximity to a promoter for a different gene. The promoter for gene A may be a highly active promoter with enhancer sequences bound by activator proteins that push transcription at a much faster rate than, say, gene B is supposed to have. And in the next example, you can see that there are fusion genes being produced, in which we have the normal coding sequence here for gene B and the normal coding sequence here for gene A, excuse me, having been disrupted. And it's a little bit faint on this image, but there's a line right here to indicate that there is a break that has occurred along this particular line. So the coding sequence for this region here on gene A is then going to be fused to the coding sequence of this gene region on gene B. And what the outcome is then is these fusion genes down below that are showing, got lots of arrows here, that are showing that we have these new sequences having been put together. This can actually completely disrupt a gene from being able to be functional, or it may generate a totally new function for the gene expression or the transcript, ultimately that can be translated into a protein doing something different than it had before. And lastly, with the breakpoint effect, what is seen is that there is some kind of a damage to a particular gene, possibly when we had a simple translocation take place where one large segment of a chromosome is moved to another, and in doing so, this large segment here has gone off into oblivion to a different chromosome, and it has changed where this total gene sequence would have been, and so now this gene down below is no longer going to be functional. We've deleted the coding region for that particular gene, and we don't have the ability for it to be able to do what it normally was supposed to do. This can be a problem leading towards the loss of tumor suppressor genes, especially. So those are those large chromosomal changes that we may see happening. But what may also occur, especially in the context of solid tumors, is that we see individual nucleotide changes or small changes within the DNA sequence or DNA alterations. All of the chromosomal changes that were just described are predominantly observed in hematologic malignancies, and smaller DNA alterations can be seen in hematologic malignancies, but these, when there are point mutations or small changes in the DNA sequence, typically that is what is found in a solid tumor oncologic disorder. So there are a number of different DNA alterations, some of which are spontaneous, some of which are induced. Spontaneous mutations are those that occur within the genome on a regular basis. We're all experiencing spontaneous mutations all the time, but we have DNA repair mechanisms to fix those mutations whenever they occur. So just in problems with DNA replication in a mitotically active cell, if DNA polymerase makes a mistake, it does have exonuclease activity to identify that mistake, cut it out, and fix it. But if it makes a mistake and doesn't actually notice that there's a mistake there, then it gets left in the genome, and that's what's considered to be a spontaneous mutation. And it can be, the categories below can be spontaneous. Otherwise, an induced mutation would be those that are exogenous to the cell. UV light, exposure to various chemicals, viruses, all kinds of different things where a cell may be exposed to something from outside of itself, and that exposure causes any of the kinds of mutations or alterations that we see below. So missense mutations, maybe you haven't thought about some of these terms in a while, just in case. A missense mutation is a base change that is going to encode for a different amino acid than normally would have been encoded for. This is sometimes not problematic if it is an amino acid that has a very, very similar structure as the original amino acid. If it's a completely different amino acid, or it's an amino acid that's in a position of that of the protein that has a very purposeful function along the lines of enzymatic activity, then by losing that amino acid, you have destroyed the function of that enzyme, as an example. A nonsense mutation is one where a single base change can change a normal codon to a termination codon. Or it may be that there are a series of base changes that changes a normal codon to a termination codon. If this occurs very early on in the protein sequence, then the stop codon occurs early on in the DNA sequence, is what I should have said. Then the mRNA is not going to be will be transcribed, but the protein ultimately will not have translation occur because it will identify that particular site as being, don't bother putting the next amino acid on, we're done here. And then we end up having an extremely truncated protein, if any protein can be made at all. So because of that, these nonsense mutations are particularly problematic when discussing tumor suppressor genes. It is when we lose tumor suppressor genes that it is problematic to the generation of cancer. Insertions and deletions can result in frame shifts, and these frame shifts change what the overall reading frame is within a particular gene and change that coding sequence. When we have these frame shifts, it is going to be that we are generating a whole new amino acid sequence because of these small insertions or deletions. Generally speaking, these have to occur in groups of 1 or 2, 4 or 5, but not in groups of 3. In considering these kinds of mutations, what I've just described are missense, nonsense, and insertions and deletions that all occur in coding sequences of genes. But there are plenty of other alterations to DNA that can occur that are not within the coding sequence. And if we have promoter mutations, splice junctions, 5' and 3' untranslated regions, regulatory elements, all of these are regions that are not encoded for in the translated protein, But if we disrupt any of these various sequences within a gene, we can drastically decrease or increase its expression, or alter and jumble up the way that it ends up being translated in the case of some of these splice junction mutations. And in either case, it is going to change the outcome for that gene. All of this leads to oncogenes and tumor suppressor genes. Oncogenes are those genes that are going to be turned on and are going to have some kind of a gain of function. They are going to be genes that are going to be very useful to promoting the proliferation of a cell by turning on a receptor that isn't supposed to be turned on, or turning on some sort of regulatory element within the cell cycle that allows for the cell to push through the cell cycle when it shouldn't normally do so. So oncogenes are going to are actually normal genes initially. They're called proto-oncogenes, and they have a normal function within the cell. It is only when there has been some kind of an alteration to that proto-oncogene that it disrupts its normal function, leading it to an oncogene and leading it therefore to this new gain of function. How proto-oncogenes can be activated are varied, a list of which is here. Viruses and chromosomal translocations, any kind of amplification of genes, or point mutation, all of these are going to potentially lead to turning on a proto-oncogene into an oncogene. And as you can see below in this lovely image, I have this little yellow cell here with green chromosomes, and there's a little green ball here that represents a particular allele of a particular gene sequence. Gain of function mutations or in proto-oncogenes are typically dominant mutations, meaning that if just one allele is altered, and we can end up seeing, and so then we end up having this one allele becoming altered by some sort of DNA damaging agent, we can see tumor formation because of just one single allele. Certainly there has to, initially being turned on. Certainly there has to be a number of different changes to the genome within a cancer cell, within a cell, for it to actually become a cancer cell. But initially, it is just a single mutation to one of these proto-oncogenes that gets the process started. An example of how oncogenes may function is through receptor tyrosine kinases. And if you haven't thought about signal transduction, it's actually kind of a useful thing to understand when trying to dissect molecular diagnostic mechanisms in oncology. So initially, when we have a ligand here in purple in the presence of some transmembrane receptors, those receptors dimerize, and they become activated by a number of different mechanisms. This is the basics of signal transduction here. That activation can then activate signal transducing molecules, proteins within the cell, that can then activate other proteins, and can activate potentially transcription factors that can move into the nucleus and bind to DNA and recruit in, with the help of some general transcription factors, recruit an RNA polymerase to start the gene expression and creation of an mRNA molecule that can ultimately be translated into a protein. It is when any of these mechanisms go wrong that we disrupt this central dogma of molecular biology. DNA can replicate itself, DNA can be transcribed into RNA, and ultimately be translated into protein. If what happens is there is an alteration in the receptor, maybe in the intracellular portion of that receptor where it is constitutively active, then we end up having this signal transduction cascade moving forward without being regulated. There are a number of instances in which any of those signal transducing molecules found within the signal transduction cascade are active without being activated by the receptor. There are certain transcription factors that might be activated without actually having activation from outside in the cytoplasm and ultimately up to the surface of the cell. So we can see multiple places where signal transduction goes awry, and it is molecular diagnostics that can be used to help identify these specific molecules that are known to be altered in a number of different cancers, to help us identify and personalize some of the medicine to be used for patients. In the context of oncogenes, I've given you a couple of tables, and I've requested that these slides be made into a PDF for your visual interest afterward, so that you can have some of this information at your disposal. Notice for what I've given you first, there are hematologic disorders. In the next slide, you can see that there are solid tumor disorders as well. But notice that the alteration that takes place for these hematologic disorders is oftentimes some kind of translocation. Certainly there are point mutations, and certainly this is a very abbreviated list, but it would take 12 PowerPoint slides to be able to put all of these into a list that you could actually look at. So this is just a short list, and these are some of the molecules that we can test for now to determine if any patient has some of these alterations and personalize their medicine. But you can see that the disorder is listed on the right. What the alteration is in the middle, and the fusion gene, or the altered gene for that matter, is present here on the left as an oncogene. Likewise, with solid tumors, there are a number of different changes that can occur, many different kinds of changes. But usually it is some kind of a point mutation or an amplification, less likely that we have translocations, although they do happen, but mostly that we see an increased production of a particular gene sequence being transcribed into an RNA translated into protein, or a point mutation which changes the effect of one particular gene. There are many different point mutations for what I have listed here. There are some more well-known examples than there are of others, but there are so many it would be exhaustive to be able to list them all. The tumors that some of these are associated with, you'll notice there are multiple different tumors that some of these oncogenes can be associated with. It depends on the source of the alteration and where that cell first exists, for which solid tumors we actually see those in. So tumor suppressor genes would be the flip side to the oncogenes. These are going to be genes that are encoding for proteins that will ultimately inhibit cell proliferation. By having some kind of a mutation or disruption of the coding sequence, what has happened is we lose or at least reduce the function of any of these tumor suppressor genes. This is what leads to cancer development for these particular problems. The Knudsen two-hit hypothesis suggests that one copy of one tumor suppressor gene can be lost, and it is essentially a recessive mutation that occurs. There's a second copy that can overcome the loss of the first copy. But when we lose that second copy, we enter into a state that's considered to be loss of heterozygosity. So both alleles of a tumor suppressor gene must be altered. We can see inherited states where we inherit one bad copy, and then there's a sporadic mutation that occurs to the second bad copy. Otherwise, there may be two sporadic alterations that occur to the two alleles of a tumor suppressor gene. This loss of heterozygosity comes about when you look at what a normal cell would look like. It has two homologous chromosomes that have all of the gene sequenced on them that are totally normal. But at one particular region, a DNA damaging event in the sporadic model would cause some sort of a mutation at this particular site. But the second copy of that allele is still functional. It's still present. So this cell, the genome for that cell, would be considered to be heterozygous. There's one good copy, there's one bad copy. So this cell is a heterozygous cell and is therefore not problematic. It is only when a second DNA damaging event has occurred, as is shown in this third cell type, that this cell is no longer considered to be heterozygous. Both copies of this allele have been altered, and therefore this cell has acquired two bad copies and is now homozygous for that particular allele sequence. So it has lost the state of being heterozygous, and we do not have that particular tumor suppressor acting in the manner that it should. And we therefore will see tumor formation. The example I gave for oncogenes, I gave you this image of signal transduction. Certainly, it's not the only way that we can see oncogenes, but it's generally speaking a pretty good model. With tumor suppressor genes, a pretty good model would be to represent the cell cycle. And any of the molecules that are involved in pushing a cell from one phase to the next throughout the cell cycle can be altered and can be problematic in leading to the occurrence of cancer in a hematologic malignancy or a solid tumor. So if you recall your cell cycle, central dogma of molecular biology and the cell cycle, good times, right? There has to be some kind of a stimulus that would be here, and that can move the cell from a resting state in G0 into G1 or gap 1. And there are a number of different molecules. I've just listed a few here, but there are several molecules, hundreds of molecules that are important for allowing for a cell to actually progress through G1 and into S. The DNA damage checkpoint shown here, or restriction 1, that particular checkpoint is very, very important, and P53 is the molecule that is probably the most important for allowing for a cell to progress into synthesis, S phase, and checking to make sure that DNA is in its perfect state. And if we don't have a perfect state of DNA, then passing into replication of the chromosomes cannot ensure that we have DNA polymerase acting the way that it should and allowing for a perfect copy of the genome in its original state. And you can see that P53 acts in many different places as well. Certainly P53 isn't the only tumor suppressor gene, it just happens to be the most well-known, but there's a few others that are listed here and several others that, that I have listed for you here. So along the lines of tumor suppressor genes, what we saw with compared to oncogenes, what we saw was there were amplifications, there were point mutations, there were translocations. Along the lines of tumor suppressor genes, what are typically observed are some kind of deletions, maybe promoter methylations, in which the CpG islands that might be found just upstream of a promoter sequence are turned off and do not allow the regulatory sequences that would allow for gene transcription to commence at the appropriate rate can be shut down. And then there might be large regions of deletions in large portions of chromosomes, all of which are going to be leading to the loss of particular genes or the change in the sequence of other genes. And the hematologic disorders are listed to the right here. In regard to solid tumors, as I mentioned, P53 happens to be a very common one, but you can see that the mutations are also very similar as what we see in hematologic disorders, in that there's lots of deletions. There's a few more point mutations that are observed in solid tumors, but there's certainly some point mutations that are observed in hematologic disorders as well. And some of the solid tumors are listed here. Again, this is not a complete list. It is just some examples of some very common tumor suppressor genes, and as we saw earlier, oncogenes that are known to be altered, and a few of the ways that we can alter them. Yes, there's point mutations. How many point mutations there for any given gene? We can know of 10 to 100 different mutations, or maybe just one. Too many to be able to list. So now what all of this leads to, now that we know how we got our genes and our chromosomal changes, now we're going to actually start looking for them. And it's molecular diagnostics that are going to help us to determine whether or not we have the presence of these oncogenes or the loss of these tumor suppressor genes, and help to determine whether or not a patient is going to benefit from any given therapy. We first have to be able to detect the presence or absence of these genes. So for hematologic malignancies, lymphoid clonality can be considered both hematologic and solid tumor, depending on who you're talking to. But I will describe a little bit of lymphoid clonality. But certainly, with the detection and monitoring of fusion genes and of acquired mutations, this is where we're definitely going to be looking at the hematologic side. But there are far more acquired mutations that are going to be seen in the solid tumor world. Ultimately, what an important point that not everybody is aware of is the concept of minimal residual disease, especially in hematologic disorders. We're able to do much less with this in solid tumors simply because of the invasiveness of being able to look for those solid tumors. In a hematologic disorder, you draw some blood or potentially do a bone marrow tap. It's still invasive, but can be far less invasive than considerably speaking, considering to biopsy a solid tumor. And then there's a few other things that we'll discuss as well. leading to this whole idea of molecular diagnostics. So lymphoid clonality refers to the presence of a hematologic disorder, or some people may refer to a lymph node as being a solid tumor as well. It depends on who you're talking to. The hematologists tend to think of all of these things circulating, and so they're going to be of the hematologic disorders. What is a normal event in lymphocytes is this gene rearrangement, and this is a very, very good thing to have gene rearrangements take place, because that allows for diversity of our immunoglobulin genes and T-cell receptor genes so that we can fight off infection. We can use the context of lymphoid clonality to help detect leukemias and lymphomas and to monitor them as they have progressed through a therapy, and ultimately look for whether or not specific rearrangements have taken place to help determine a prognosis. My little picture here up above showing all of these different colored circles represents a polyclonal cell population of lymphocytes. And in any given human at any given time, generally speaking, we would see a varied population of cells, because we've got all of these different cells with different rearrangements of anaglobulin genes and also of T-cell receptor genes. In the context of a leukemia or a lymphoma, what ends up happening is we end up seeing many, many, many copies of one particular gene rearrangement being expressed on the cell. Clonality does not necessarily equal malignancy. However, when you see a large population of cells that contain the exact same gene rearrangement, it certainly makes one take pause and wonder if this is actually a clone of lymphocytes that is going to be problematic for the patient and leading to a cancer situation. How to test for that lymphoid clonality would be to identify very specific regions that are known and use primers specific to those regions. So in looking at anaglobulin genes, we know of the variable diversity in joining regions, and then a few other gene sequences, the IgH, K, and L. And then there's also the T-cell receptor loci, leading to the various different chains that will generate a T-cell receptor. The image that is shown here is a polymerase chain reaction followed by gel electrophoresis, capillary gel electrophoresis, and I'll briefly explain that in just a moment. But in a normal polyclonal population, an individual that does not have a leukemia lymphoma, then what we see is this all of these different little peaks that are associated with many, many, many different gene rearrangements, in this case of the IgH gene. And down below here, this is just one example looking at the T-cell receptor gamma chain, and you can see that there are many, many different populations of cells with different rearrangements of that gene. Looking at a clonal population, you see one peak. So that would be representative of that previous slide where we just saw all these blue balls, essentially blue circles, representing an accumulation or a clone of those lymphocytes. And this is problematic because it's suggesting that we have far too many cells that have the exact same gene rearrangement, and therefore there is a mitotically active precursor that has gone a little bit wild and is making many too many copies of itself, and what is the outcome there is a clone. How these images came about is through a polymerase chain reaction, or a PCR, in which the sequence that is to be identified must be known, and so that we consider that the patient's template, their DNA. There have to be primers involved, DNTPs, the individual A's and T's, C's and G's, Taq, polymerase, and certainly some buffers. But all of this has to come together, and we would then melt this double strand of DNA, allow for a primer to bind to a specific region that we know that we're trying to assay. And then in the slew of all of these other components, then Taq polymerase can extend that region that we've allowed a primer to bind to. Taq polymerase must recognize double-stranded DNA, which is why we need to have a primer first, and then it can extend and move onward, outward, and extend out this sequence, and we can get many copies of that. And if you haven't thought about PCR in a while, you can start off with one copy of your template DNA and have an exponential number of copies depending on how many cycles that you allow for the reaction to take place. In capillary gel electrophoresis, there is actually oftentimes polyacrylamide that is going to be found inside a very, very, very skinny capillary tube. And it is, the DNA is allowed to, after it has gone through a polymerase chain reaction, the sample is then passed through this capillary tube and through a current. And where these sequences end up within the tube, we can then measure and detect where these different peaks occur. In the detection of fusion genes, so that was lymphoid clonality, and moving on toward the detection of fusion genes. These fusion genes are going to be very much associated with the activation of proto-oncogenes or the generation of new oncogenes. Oftentimes, the detection of these fusion genes, of these proto-oncogenes, is going to be performed by using real-time PCR, or also known as real-time reverse transcriptase PCR. And it is these fusions that we are trying to identify by using this particular method. A reverse transcriptase real-time PCR can be followed in real time, but what first has to happen is we have to generate an RNA, identify an RNA sequence from the sample. This is kind of tricky because RNAs are very easily degraded, but in getting one of these RNAs, a primer to that RNA, oftentimes at the poly A tail of that RNA, is going to be exposed to an enzyme called reverse transcriptase, which will then create an RNA-DNA hybrid of the original RNA molecule. The RNA can then be degraded. So this RNA portion can then be eliminated in a number of different ways using an enzyme called RNase H or using high alkali, and allow for a primer to bind to the single-stranded DNA that remains. And in the presence of the enzyme DNA polymerase or Taq polymerase, we can then generate a double-stranded copy of DNA. That double-stranded copy of DNA is going to be detected by a number of different assays. There are a number of different dyes that can be used to detect whether or not a particular sequence of DNA happens to be present from that original mRNA molecule. So cybergreen and evagreen are two very commonly used fluorescent molecules to detect that. But more frequently, what's used are hybridization probes. And the most common of which is the, sorry, my arrow's kind of, I'm supposed to hit undo here. My arrow's a little off. The TACMAN assay is the most commonly used real-time PCR assay, but there are certainly several others that I've listed here. So essentially what happens is there's a probe that is going to be attached to the DNA molecule, and that probe is going to have two ends, one of which contains a fluorescing molecule and a quenching molecule. And when that probe is allowed to bind to the DNA, in the presence of DNA polymerase, it will be displaced or chopped up, depending on which assay is going to be used. In either case, if the probe has bound to a very specific sequence and is then removed, the fluorescent molecule is then able to actually fluoresce in the presence of light, and the quencher is no longer in close proximity to allow for the blocking of that fluorescence. And so we can see in a real-time PCR assay or a real-time reverse transcriptase assay, we see fluorescence, and we see many, many copies of fluorescence, and we can see an increase in the fluorescence depending on how many copies of that particular gene sequence we have. This is a commonly used mechanism to detect the BCR-ABL fusion protein from the translocation of 922 in CML, chronic myelogenous leukemia, and there are a number of different common variants of that particular fusion that can be identified. It's also used in the hematologic setting to look for BCL2 IgH translocations from the translocation of chromosome 14 and 18 in follicular lymphoma. It is certainly used in some solid tumor malignancies as well, but these are very, very commonly used in these two particular malignancies. Essentially what happens is, as the fluorescence is generated, there is a threshold that must be crossed, and upon crossing that threshold, we can quantify how many copies of a particular gene sequence we are seeing in an individual. And then that can be converted back into a standard curve, and we can get an actual number of copies for a particular gene sequence. To detect fusion genes in another context, we might see the use of karyotyping, which is the sort of standard for looking at chromosomes to look for large additions or deletions or translocations. Or what might be used is FISH, fluorescence in situ hybridization, in which we are looking for structural changes by using some of these fluorescent markers. The FISH is a very specific probe, as opposed to karyotyping where you're looking at the whole genome. You're using FISH really to look for very specific sequences. And what's used is a fluorophore, a DNA probe that has a fluorophore that can bind to a complementary sequence of DNA and then fluoresce once exposed to light. And it will only fluoresce if it is bound to a very specific sequence. So in this case, it would need to be known that there is a likely translocation that has taken place, and you're looking for that fusion gene that would have been created because of that translocation. One nice feature about FISH is that we can also use dual fusion FISH to look for two different colors or two different fluorescent markers. In the case of BCR-ABL, we can look for one gene being encoded for in green, the other encoded for in red. And if there is a translocation that has taken place, then you can see that there is a layover in what is observed as yellow because of this overlapping of the red and the green fluorescent probes. And we're actually being able to identify that fusion gene because of these probes that bind to very specific sequences on these large chromosomes. Where we're using FISH is most commonly in hematologic malignancies, but it can be used in some solid tumors as well. With breast cancer and looking for HER2/neu, FISH can be used as well as a number of different immunohistochemistry methods as well. But typically, if we're talking about a molecular diagnostic tool, we're trying to look for the DNA, not the protein product. And then immunohistochemistry is looking for the protein product. So by using FISH, there are many different hematologic malignancies, fewer solid tumors that are actually going to be using that. But those are all listed here for your reference. Why FISH is useful? Certainly it's because in karyotyping, you have to have metaphase chromosomes. And with FISH, you can use interphase chromosomes. You don't have to have cells that are able to divide. So you can do this on archived material. That's a very nice benefit. It's much faster, and it's much more sensitive than we can see with using karyotyping. And using two colors, we can get much better discrimination. It might be useful to use FISH initially to determine whether or not we have the potential for a translocation having taken place. But real-time RT-PCR is going to, real-time reverse transcriptase PCR is probably going to be used later on as a confirmatory mechanism. There are commercially available FISH probes for all known translocations, but certainly there's other translocations that we don't know about. Some problems with FISH is that they are informative to very specific regions, where if you're using cytogenetics and karyotyping, you can look at the whole genome. And so they are currently only being used as an adjunct to cytogenetics. You have to have a certain number of cells that must be positive to be able to detect using FISH any of these changes, and it is not as sensitive as is real-time PCR. We don't actually see allele-specific changes, and you can't detect any small changes. You're having to look for larger chromosomal changes when you're using FISH. So along the lines of some of these solid tumor malignancies, there are going to be plenty of mutations within genes that are not just going to be associated with translocations. And many of these mutations are going to be changing oncogenes or proto-oncogenes. There are so many different methods, I could not possibly go through them all. I've listed a few of them here, and these can be used for both hematologic or solid tumor malignancies. One of those is allele or mutation-specific PCR, in which there are mutant primers that are bound to a mutant sequence, or primers bound with a sequence that will bind to the wild type sequence. And depending on whether or not one of those primers binds to the sequence or not determines whether or not a particular band size will be observed after performing a PCR and a gel electrophoresis. So you can see that the larger fragment size fragment band is observed when you have the mutant primers there, as opposed to a mismatch with the wild type primers. Likewise, there are mechanisms to use real-time PCR for these single mutations, as opposed to looking for just translocations as we saw previously. Again, it is in using these fluorescent probes that are containing these sequences that contain both a quencher and a reporter, and whether or not that quencher is going to be displaced in the presence of these probes. So having a wild type allele bound with a mutant probe, or a mutant allele bound with a wild type probe, you have to know what you're using, can determine whether or not you're going to be seeing fluorescence. This is commonly used with the JAK2 mutation that's associated with many different myeloproliferative neoplasms. And it's also used with several others. BRAF is a common one in melanoma and solid tumors that can also be used. Again, the fluorescence has to cross a threshold, and you can actually quantify how many of those gene sequences you have. High-resolution melt analysis is going to be the incorporation of these probes, and then allowing them to bind, and then seeing how long it takes for them to dissociate from the gene sequence. If you have a perfect match, it will take much longer for the probes to dissociate, and then it will take longer for you to be able to detect the fluorescence from that dissociation. So you can compare wild-type sequences to mutant sequence to detect whether or not a patient contains either the wild-type or the mutant. Minimal residual disease is one of these prognostic mechanisms that is very, very useful in hematologic disorders. And it's useful to determine whether or not a patient has any cells left over after having been treated. The name says it all, minimal residual disease, what's left over after treatment. So any of the tumor cells that might still be circulating after the curative treatment, and these are very, very useful for predicting whether or not a patient has relapse, or whether or not a therapy is working. So since these are with circulating cells, they can be less useful in solid tumor malignancies, except for when those tumors have metastasized to distant sites. If you are detecting those solid tumor cells in the peripheral blood, then likely you are still seeing minimal residual disease. Where this theory comes from is the cancer stem cell theory, and that there are cells that act as progenitor-like cells, that act as terminally differentiated cells, and that are going to maintain some characteristics of those within a cancer cell. So when we give an individual some sort of a therapeutic regimen, then we can see that the vast majority of mitotically active cells are going to be eliminated. But there are some resistant cancer stem cells that are likely to have escaped that therapeutic regimen, and therefore that's what it is that we are trying to be able to detect. This has been described in breast cancer cells after conventional treatment, and has also been described in a number of different hematologic malignancies, especially the 5q- myelodysplastic syndromes, in which there are specific cell types that are resistant to chemotherapeutics and remain behind, so that we say a patient has maintained this state of minimal residual disease. Those tumor cells can be detected in a number of different places, in the blood especially, but certainly in some body fluids as well. Using traditional imaging and biochemical methods, you can detect 1 in 100,000 non-tumor cells, but using some of the molecular methods, you're able to detect 1 in 10 million non-tumor cells, so it's far more sensitive. But you have to have some kind of a cancer-specific marker, a leukemia-specific marker or a solid tumor-specific marker that is found in all of the cells, and it's going to remain within all of those cells. And it should also be a marker that's unique to each individual. And then you can use several of the methods that we've already discussed to detect that. Post-transplantation chimerism is essentially for those individuals receiving bone marrow transplants, and it is looking for the gene sequences that are found in some of the donor cells, found in the recipient cells, and looking to see whether or not we end up having a mixed chimera for one of these individuals. What is oftentimes being tested for are some short tandem repeats or some other loci, but oftentimes these short tandem repeats, which are 3, 5 base pairs, sometimes a little bit longer, that are repeated over and over and over again in one short region. What can be done is a polymerase chain reaction followed by capillary gel electrophoresis to look for these various peaks. On the top, you can see that we have a recipient that has 2 different peaks than does the donor. So after we can see that the donor then has these 2 alleles being informative alleles, that we know that we're going to be looking for those in the recipient after transplantation. For this individual at the bottom post-transplantation, we're seeing all 4 alleles, which means that there are still some of the patient cells remaining as well as the donor cells, and it really needs to be just the donor cells that are going to be present if we know that a bone marrow transplant has taken place. So this is just one other tool that we can use molecular diagnostics to be able to test that. Sorry, my screen went blank for a moment. Where some of molecular diagnostics is moving to is whole genome diagnostics as well, and looking at using microarrays to be able to detect expression levels of genes, so that some of those non-coding sequences that we were talking about earlier that can be problematic for genes can be observed in some of these microarray technologies. There are going to be very, very small sequences of DNA that are going to be affixed to a chip. These are very, very small, and thousands of different gene sequences can be assayed, becoming relatively cheaply for detection as well. Single nucleotide polymorphisms of known mutations can be assayed, as well as looking for copy number variants. So we can see if we have some cells that have many, many, many different copies of an oncogene, that is what is leading to the proliferation of those cells. We can also use comparative genomic hybridization to be able to detect some of the larger chromosomal changes or copy number variants. Remember, with we have unbalanced this is only going to be seen when we have unbalanced chromosomal changes, because we wouldn't see an actual change in the copy number if we had some of those other translocations that would be observed. So what's actually being detected for is extra copies of a gene sequence, and this is very commonly observed in certain disorders. I've just listed a couple of here. There's actually several more that could be included, but in hematologic malignancies, the IKZF1 gene can be observed in childhood acute lymphoblastic leukemia. It is absent, and so we would see a change in the copy number compared to normal. And in solid tumors, we can see additions, duplications, deletions in various chromosomes. What's observed over here on the right is that there are actually fluorescent markers that are going to be aligned with and attached to the chromosomes, and then there's going to be detection compared to what would normally be seen, and there will be an increase or a decrease in the fluorescence of these particular gene sequences, whether or not we have a duplication or a deletion. All of this leads to the diagnostic mechanism of cancer is moving much, much more toward a molecular genetic-based mechanism of testing for these changes in the DNA, as opposed to using cytochemical stains or immunohistochemistry. The technology has moved much more towards molecular diagnostics. We can use these tools to diagnose patients if they have this particular lost tumor suppressor gene, they've lost heterozygosity. The people in the laboratory are going to be, in molecular diagnostics, are going to be doing a lot of p53 loss of heterozygosity testing. We can use that to help diagnose a patient for some of the gene sequences that might be fused together. We can also use that to help give the prognosis for a patient. A patient that has a normal p53 status one day and then a few months later has lost that heterozygosity, we're going to be using that as a prognostic factor to determine that they're not going to be doing so well. We can monitor therapies and the disease process, and also help to detect whether or not we actually have minimal residual disease or a low level of residual disease in any of these patients. Some of the high throughput whole genome sequencing approaches are becoming far more common in practice, and you'll be seeing many, many more of those processes and technologies in the near future as well. I will gladly take some questions at this point, and I see that I have a few on the side. There's one question: Are the single point mutations the same as SNPs, or single nucleotide polymorphisms? Some people do tend to use those two terms interchangeably. A true polymorphism is not necessarily something that's gone wrong in a gene. In some instances, some single nucleotide polymorphisms can change the way that a protein product functions significantly enough that it can lead toward disease. But a lot of people end up using SNPs or single nucleotide polymorphisms and single point mutations interchangeably, when in actuality they're not entirely interchangeable. Usually when I'm thinking of a single point mutation, I'm thinking of the DNA sequence and one specific nucleotide has been altered. A polymorphism usually suggests that it's an inherited change, which certainly can lead to cancer as well, but usually a polymorphism suggests that it is just a slight change in the activity of the protein function. The next question is, what is the best way to identify the fusion transcripts in a tumor sample? Well, that's a good question. Essentially, it comes down to what exactly it is that you're looking for. It's kind of common practice for particular genes to be detected in particular ways. It depends upon what assay the laboratory happens to be using and what instrumentation the laboratory happens to have on hand, or if it's in a research setting, you could be doing any number of these different assays. If it's a fusion transcript, FISH is very, very useful if you know what that sequence is, because you have to have a probe for that known transcript. Real-time PCR is very useful as well if you have a known point mutation. Is one better than the other? In some cases, yes. FISH is going to be useful if you have a fairly large number of cells. Real-time PCR is more sensitive. You need fewer cells. And you are going to be looking at the single nucleotide level, whereas FISH you're going to be looking at a larger transcript. But what happens with real-time PCR with these fusion transcripts is you're bridging where that fusion has actually taken place, so that you can see the actual transcript or maybe loss of it at another place within the genome. So what's the best way? It depends on what you're looking for. If you're screening patients, you should probably be trying to use FISH if you know what that transcript is that you're looking for. If you're looking for a very, very sensitive assay, real-time PCR might be the way to go. And certainly you don't even have to use some of these more high-tech techniques. You could also be just looking for allele-specific changes as well by using some simple PCR. Oh, here's a comment. Mutations have lower than 5% frequency in the population. SNPs have a higher frequency of over 5% in the population. That's a very good point, indeed. A mutation, which is why I say that some people use these two terms interchangeably, when in actuality they aren't really interchangeable to be using, but a lot of people do use them as such. Point mutations and SNPs, people tend to use together. But I agree with you, exactly, that a mutation would show us less than 5% frequency, whereas a SNP is something that's a little bit more common that we're going to be seeing at a higher percentage. Do I have any other questions? Well, I will then take you to this last slide that allows you to get your continuing education credits. I very much appreciate your interest in this talk, and if you have specific questions, I'm sure my contact information is somewhere within the world of BioConference Live, and I will happily take any emails from anybody that is interested in any additional information. And that's all. Thank you very much.